All right, so we left off last class describing the mechanism of transcription. Um, and we talked a bit about how it relates, at least we introduced diabetes, and we'll wrap it up talking about how it relates, how transcription, and particularly the mechanisms that regulate transcription relate back to diabetes. And we'll also be applying that as well to anabolic steroids today. Either through the readings that you guys have done for your projects, or whenever you've been in a lecture that talks about gene expression, or if you've just heard mention of gene expression in the past. Usually what that comes down to is some degree of regulation of transcription. We usually, when we talk about gene expression, that means that that gene was transcribed, and there are key regulatory mechanisms that control that gene expression. And that's usually what's oftentimes underlying this gene expression that's talked about either in the papers you read or in the other instances you've heard that phrase. And so a lot of the protein expression that you see is going to be primarily controlled at the transcriptional level as a result of the mechanisms that regulate it. One of the forms of diabetes that we touched on really briefly last class is MODY, mature onset diabetes of the young. And it's a defect in gene regulation of insulin. And so it's not exclusively just the process of transcription, but rather how transcription is regulated. And in addition, anabolic steroids act by modifying or altering the regulation of gene expression. Anabolic steroids, when you hear anabolic or anabolism, that usually means building up or the synthesis of. Catabolism usually means the breaking down of. And so when you hear anabolic steroids, that typically involves the use of some steroid that is an analog or a synthetic form of testosterone or something similar to it that leads to anabolism, the building up of protein in tissue. As you can imagine, there are a lot of side effects associated with anabolic steroid use. There are far too many side effects that are associated with it. If I was to try and break it down really briefly, it leads to dysfunction of reproductive organs, can lead to overall organ failure, can lead to psychological issues, cardiovascular issues, and a variety of different, and can lead to a lot of altered susceptibility to other diseases or infections as well. This is distinct from testosterone replacement therapy. And so that's usually focused on trying to get testosterone to a physiological level, whereas anabolic steroid use is typically trying to get testosterone levels to a super physiological level, above normal levels. And so again, there's far too many things for us to go into great detail here, but summary is, please don't use anabolic steroids. So why do athletes typically use them? Typically leads to increased muscle mass, also leads to loss of fat tissue, increased strength, reduced recovery time, and in some cases, for some competitive athletes, increased aggression that can aid in their performance. Therapeutic uses typically of anabolic steroids or of some sort of analog of testosterone or of some sort of steroid can typically be as a result of postmenopausal declines in bone density or in libido. It can typically also be used as a treatment for anemia or inflammatory diseases. And again, or just largely can sometimes be used to try and restore physiological levels of sex hormones. And so we're going to revisit anabolic steroids or steroids in general at the end of the lecture when we kind of go over all of the mechanisms of transcriptional regulation. But we're now going to pivot and switch into looking at gene regulation first within prokaryotes, because there's a bit of a model there that will help kind of set the stage for our conversation around transcriptional regulation today. In some instances where there's not sufficient glucose available, glucose being the main energetic substrate that prokaryotic and eukaryotic cells use, When there's not enough glucose, there are sometimes alternative energy sources or fuel sources that are available. One of them being lactose. And so lactose is typically shuttled inside the cell through galactoside permease. And once it's in there, there's an enzyme that breaks lactose down. That enzyme is called beta galactosidase. It breaks lactose into glucose and galactose. And once it breaks it down, glucose can now be used and it's available as a substrate for energy to synthesize and create ATP. And so again, I briefly defined anabolism versus catabolism. This is an example of catabolism, taking lactose and breaking it down into its constitutive parts, enabling glucose to now be available and used as a substrate for energy. It'll come back eventually. So for the use of lactose to be used here, there needs to be galactoside permease to shuttle it into the cell and beta galactosidase to break it down into its constitutive parts. The expression of these two enzymes actually kind of work together. And there's a model that's used to try and describe how genes are expressed and regulated in prokaryotes. That's called the operon model. An operon is typically a set of genes that have a related function with one another that are encoded together in one single piece of mRNA that then are used to translate and synthesize the proteins. So I'll try and describe this using different words. The operon model, an operon, is a set of genes that are transcribed together into a single mRNA strand and then synthesized together to produce their protein counterparts. And so what that means here is that there's going to be a number of genes that are transcribed together. They're going to be produced together in a single mRNA strand, but then used, that mRNA strand, once it's transcribed, is going to be used to synthesize their proteins. And so that means there's going to be typically a 1-to-1 ratio because they're regulated or transcribed together. So what that means is if there's multiple genes within one transcriptional unit here, which is typically the case in the prokaryotic system, we call that being polycistronic. Poly meaning many, them being together on one single transcriptional unit. In the eukaryotic system, we're typically monocistronic. There's usually one gene in a single transcriptional unit. In the prokaryotic system, they're polycistronic. And when the functions of those genes, of the proteins those genes encode for, if they're related with one another, they're typically a part of what we call an operon. And so this right here, what's depicted, it's called a lac operon. And within this lac operon, we have the genes for beta galactosidase and galactoside permease. And so they're encoded together and typically will end up being expressed together when lactose needs to be used as a substrate for energy. Excuse me. And so we have a few key, actually, I'll jump ahead to the next slide. And so this operon is going to consist of a few key parts here. We have the structural genes, which are actually what encode for the enzymes and proteins of interest that are particularly important for this operon. Then we also have the regulatory genes. And what I should say, sorry. So in the case of the lac operon, the structural genes here are the LAC-Z gene for beta galactosidase, the LAC-Y gene for galactoside permease, and the LAC-A for transacetylase. We're not going to touch on transacetylase here, but beta galactosidase and galactoside permease are the main things we're discussing in the lac operon. And so those consist of the structural genes within the lac operon. There's also going to be regulatory genes that are within an operon. Those are going to be helping to control the expression of the structural genes. And so, for example, here we have a segment labeled in yellow, forgive me if I'm wrong, called the operator. That's going to be a regulatory region that we're going to talk about for a couple slides that regulates the expression of the structural genes, forgive me. And so that operator is typically located between in the case of the operon model, is going to be located between the promoter in that first structural gene. And so proteins are going to be able to bind to that operator and either promote or inhibit transcription. And so there can either be the inhibition of transcription based on proteins binding at the operator, or promoting transcription, promoting the expression of the proteins associated with these structural genes. And so what that looks like here is that there's going to be some other regulatory gene. I'm going to start wandering away from the computer, so forgive me if I'll try and raise my voice. We have some other gene, this regulatory gene, that encodes for this repressor protein. What this repressor protein does is that it can go over and bind at the operator, and based on the name being repressor, it can inhibit transcription of the lac operon here. And that's going to be largely the case for a lot of the operons that you come across at their the operator sequence. So what this looks like is, again, this repressor protein, it's encoded by a repressor gene that's not part of the operon. It's on another operon or another gene outside of this, but it's encoded there. And then the repressor protein will bind to the operator, and here it will promote negative regulation of transcription. And when it is and when the repressor protein is altered, it will no longer be able to bind, leading to transcription of this lac operon. And what that regulation and so when the excuse me, when the repressor protein is on the operator, it'll physically block RNA polymerase from binding to the promoter and inhibit transcription that way. It'll physically block the promoter by sitting there at the operator sequence. The way this repressor protein is regulated or controlled is it's actually regulated by the presence of lactose. So in the absence of lactose, this repressor protein is in its active form, where it can bind to the operator sequence on that lac operon. However, when lactose is present, lactose will bind to the repressor protein, allosterically regulate it to change its conformation, and render it in an inactive form. So what that means is, when there is lactose available, it will inhibit this repressor protein and enable transcription of the lac operon. And so when it enables transcription of lac operon, that means we can now express beta-galactosidase, galactoside permease, and we'll get expression of that transporter, we'll get expression of the enzyme that breaks down lactose to produce glucose. And so in this case, again, we'll have lactose binding to the repressor protein. It's now inactive. Because of that, it's not going to bind to the operator sequence. RNA polymerase can now come in and dock on the promoter and lead to transcription here. And we'll have expression of our beta-galactosidase to break down lactose, and galactoside permease to shuttle it in. And so this is important for the ability of bacteria to be able to use lactose as an alternative energy source in the instance where there's not enough glucose, or when there's both insufficient glucose and lactose available as an alternative energy source. And so anabolic pathways are also controlled in some of the same processes, but not literally exactly the same, but it's the same balance of regulating a regulator that dictates whether transcription can occur. And so another operon that is popularly used as an example here for transcriptional regulation is tryptophan. And so we talked about with the lac operon that it is an example of catabolism, breaking down the lactose molecule in into its constitutive parts. There is a operon for the amino acid tryptophan, where the operon leads to the anabolism, the building up using the constitutive parts to create tryptophan. So that operon is called the trip operon. What ends up happening here is that we have tryptophan synthetase, which is what's encoded in the trip operon. Each of these encode for a different part of the enzyme. Each of these, let me be more specific with my language, forgive me. Each of these structural genes encode for a piece of the tryptophan synthetase enzyme that builds up and synthesizes tryptophan. And we have a regulatory gene for this trip operon that creates this repressor protein that, again, binds to the operator sequence and regulates transcription of the trip operon. The regulation is a little different because what we had before with the lac operon was the substrate inducing expression of the lac operon. Here, we're creating an end product being tryptophan, and tryptophan itself is what regulates the repressor protein. When there's sufficient expression of tryptophan, it binds to the repressor protein. But this time, when repressor protein is bound by its substrate, in this case tryptophan, it activates the repressor protein. So here the repressor protein is activated when it's bound to tryptophan. Now it can bind to the operator sequence and inhibit transcription of the trip operon. So what that means is that the end product here, the end product being tryptophan, is able to repress transcription of the trip operon. So we had substrate induction with the lac operon, and here we have end product repression, meaning what is produced by this operon will end up leading to the repression of it. And so when there's sufficient enough tryptophan synthesized, it'll then shut down transcription of the trip operon. So just to summarize all of this, the lac operon, the substrate being lactose there, binds to the repressor protein, renders it inhibited, and now the RNA polymerase can bind to the promoter and transcribe the lac operon. With the trip operon, we have the repressor protein for that operon naturally inactive, but once there's enough tryptophan synthesized as a result of the enzyme the trip operon encodes for to produce tryptophan, once there's enough synthesized, that tryptophan binds to its repressor protein, now activates it, and suppresses and inhibits transcription of it. So there's different kinds, but both include negative forms of regulation here. I'll stop for a couple of questions on these two operons or the operon model in general. I know there's no college basketball on today. Yes. They both include, so when I say they both include negative forms of regulation, what that means is the lactose will inactivate the repressor protein. And then in the case of the trip operon, tryptophan will activate the repressor protein, negatively regulating the trip operon itself. Does that make sense? Yeah.
So, lac operon, when lactose is present, it like deactivates the repressor.
It deactivates or inactivates the repressor. Or negatively regulates the repressor.
Okay.
So it's like a double negative. It's negatively regulating this negative regulator. So double negative equals positive. So it leads to transcription of the lac operon. And if you think about it, based on what the lac operon encodes for, it encodes for what shuttles lactose in. It encodes for the enzyme that breaks it down to create glucose. So if you think about what should happen in the presence of lactose, you'd want that lactose shuttler to be expressed. You'd want the enzyme to be expressed. And so there's intuitively, at least for the lac operon, it intuitively makes sense if you think about what needs to be expressed in the presence of lactose. Does that make sense?
But isn't that it's actually critical to
So galactoside permease, which is what shuttles lactose into a prokaryotic cell, and beta galactosidase, which is what breaks down lactose into galactose and glucose. And so we want that glucose. So now that is how we can take lactose and use the glucose that comes from it as an energy, a source of energy for the prokaryotic cell. Does that make sense? All right, we'll go ahead and press on. So I've kind of I've kind of I've tried to be careful with this, but I've inferred this a couple times in the previous slides we were just on. The it's not just the presence of lactose that drives this pathway, but there also needs to be there is a requirement for needing lactose, it being the only energy substrate available, meaning there needs to be lower glucose, an absence of glucose to want to divert and use lactose as an energy substrate. And so whenever there is glucose available, these glucose pathways are what we call constitutively expressed, meaning they are always on. They are always being expressed because every process within a prokaryotic cell, as well as a eukaryotic cell, requires using glucose as a substrate for energy, and that glucose leads to the production of ATP. And so many of our fundamental cellular functions rely on using ATP as a source of energy. So these glucose pathways are, again, we say constitutively expressed or always expressed. And so if glucose is present, these other energetic pathways that use alternative energy sources are typically, for lack of a better phrase, turned down. They're not expressed nearly as much. And so that includes the lactose pathway. So in the presence of glucose, these other pathways are not nearly turned on or not nearly expressed as much. However, in the absence of glucose, that's typically where these other pathways are, for lack of a better phrase, turned up. They're expressed a lot more so that those other energy sources can be used to enable these basic biological functions to produce ATP, or sorry, to produce a glucose that can then be used to produce ATP that's required for so many basic functions in the cell. And so what that looks like is typically there's this balance between glucose and a secondary messenger that we've talked about already, cyclic AMP. In instances where glucose is high, cyclic AMP is going to be typically low. And vice versa, when glucose is low, cyclic AMP will be high. Why that's important is because cyclic AMP can form a complex with a protein called the catabolite regulation protein, or CRP. CRP and cyclic AMP together form this complex that actually serves as a transcription factor binding at the CRP site, just upstream or to the left of the promoter on the lac operon. And so when, again, the need to use lactose as an energy substrate typically coincides with glucose being low. So when glucose is low, that means cyclic AMP is going to be high. And now it can form this complex with CRP that acts as a transcription factor, enabling the recruitment of RNA polymerase to then transcribe the lac operon. And so because it's a transcription factor, this CRP cyclic AMP complex, it more readily recruits RNA polymerase to bind to the promoter and promote transcription of the lac operon. And so what can be helpful is to think about this balance of lactose expression, the presence or absence of lactose, and the presence or absence of glucose. And so if there is glucose and if there is not lactose, there would not be expression or transcription of the lac operon. And so these are going to be helpful questions that can help you kind of assess your knowledge of the lac operon and this relationship with glucose and cyclic AMP. But these would be an example of quiz questions that could come from this content. And so, are there any questions about that degree of regulation with glucose levels and cyclic AMP levels? Yes.
like would lactose still be, would they both be expressed or would it just be?
When you say would they both be expressed, do you mean?
Would like the lac operon be operating, and the glucose pathway would also be operating?
What do you think?
Yeah, probably.
Yeah, probably meaning that both pathways would be expressed.
Yeah, I figured. I don't see anything saying that if glucose is on, it's activating lactose lac operon.
Go ahead. Did you want to tack on or?
Wouldn't it be like, you said glucose and cyclic AMP go vice versa of each other?
Mhm.
And so in that case, since glucose is present, you wouldn't have the production of cyclic AMP, so you wouldn't have the production of lactose or production of.
Well, if there was low cyclic AMP, would you have no lactose, or would you have, or rather, would you not have that transcription factor, CRP, cyclic AMP?
You wouldn't have the transcript.
Mm-hmm. And so if there's still, like you were saying earlier, remind me your name one more time. Thomas. Like Thomas was saying, because glucose is there, those constitutively expressed pathways will be on. However, lactose is there. So if lactose is there, what does that mean for the lac operon? It's transcribed, it's expressed. And so you'll still have lac operon being transcribed. You'll have those glucose pathways being constitutively expressed. And so you'll have both. You won't have the lac operon transcribed as efficiently or as quickly because you don't have that CRP, cyclic AMP transcription factor. However, both pathways would still be on, just to a different degree. Yes.
It's not just either off or on.
Yeah, it's not. Rarely are things in science like binary, like on, off, yes, no. It's usually more a matter of degrees. If that makes sense. Yes.
But wouldn't you require transcription factors to
Well, there's one kind of transcription factor that is required. Yes. A basal transcription factor is required. More transcription factors binding, for example, like CRP, cyclic AMP, that will enable or facilitate transcription to happen more easily, more readily, and more quickly. However, in the absence of other transcription factors, as long as that basal transcription factor can bind, as long as the operator sequence doesn't have a repressor protein there blocking the promoter, transcription can still occur. It's just not going to happen nearly as quickly or as readily as with multiple transcription factors coming in and binding. So a few more things. It's going to be helpful for us as we finish up this lecture to define a few key terms that either you may have heard before, or at least to help try and give us our bearings before we jump to the next few set of slides. There are regions in and around the promoter, for example, like the operator sequence that we just talked about, that can dictate or regulate proteins coming in and binding. We typically call those cis-acting elements. Excuse me. Whenever you hear elements, immediately think DNA. Whenever you hear factor, immediately think a protein that acts on the DNA. Okay? And so these cis-acting elements, bless you, it's going to differ between the prokaryotic and eukaryotic system. We're going to start pivoting to start talking primarily about the eukaryotic system. So when we talk about cis-acting elements, that's oftentimes going to be on the same chromosome. And so the alternative would be regulatory genes that are called trans-acting elements because they typically exist far away. And when we say far away, we mean on a totally separate chromosome. And so these trans-acting elements, element meaning it's DNA, it's a gene, these trans-acting elements will typically encode for some protein that will act on a cis-acting element. So I'm going to take a step back. So these trans-acting elements that exist on some other chromosome encode for something like a transcription factor. So transacting elements encode for a transcription factor. This is encoded on some totally other chromosome than what our like reference, what gene we might be focusing on. But that transcription factor that's encoded on some other chromosome on a transacting element, that transcription factor comes in and binds on a cis-acting element adjacent to the promoter of our gene of interest. So cis-acting elements are usually on the same chromosome with respect to our gene of interest here. Transacting elements are typically, and again, thinking about the eukaryotic system, typically are on some totally other chromosome. However, that cis-acting element typically, or that transacting element will typically encode for some transcription factor, some regulatory protein, typically a transcription factor that will come in and act on the cis-acting elements of that are relative to our gene of interest. And so a couple of examples of this. This also these basic ideas or mechanisms are also true of the prokaryotic system as well. And again, these transacting factors or transacting proteins are typically going to be transcription factors that act on and bind to the cis-acting elements of our gene of interest. And so that's going to be either adjacent to our promoter, at our promoter, or some distance away on the DNA that it can, again, we're in three-dimensional space, our DNA is bent. It can still act on our gene of interest, even if it's further down the chromosome. And we're going to talk about like that degree of distance in a moment. Oftentimes in the eukaryotic system, these cis-acting elements are oftentimes much further away than what we were just describing with the prokaryotic system. The prokaryotic system, these cis-acting elements are usually just like adjacent. We have the operator sequence. We have that CRP site that CRP cyclic AMP can bind to. Those are cis-acting elements that are really close to the promoter in the prokaryotic system. Eukaryotic system, it's typically much further down on the chromosome. And what's important to note is that for the transcription factors that bind and act on these cis-acting elements, it can any single transcription factor can bind to multiple different cis-acting elements on genes, and they're going to be typically able to bind and recognize multiple different kinds of cis-acting elements. And on a given gene, there might be multiple transcription factors that bind to the cis-acting elements that are associated with that gene. So I'm going to repeat that again because that was kind of that might have been hard to follow. A single given transcription factor can bind to multiple cis-acting elements. There are some websites that have done some incredible bioinformatic work to help us identify what genes transcription factors are associated with. So there's a great website called UniPro, UNIPROT. If you look up your favorite transcription factor, because I'm sure by now all of you have your own favorite transcription factor. If you look up a transcription factor of interest, and if you scroll down, you'll be able to see what genes that transcription factor has been validated and confirmed to bind to the cis-acting elements of those genes. So a single transcription factor can bind to the cis-acting elements of multiple genes. If we look at a single gene, again, your favorite gene or your gene of interest, there might be multiple transcription factors that can bind to that single gene. And so as an example here, we have a single eukaryotic gene of interest that is going to be looked at in response to a couple of different stimuli. In response to stimulus A, we have this transacting element that is on a separate chromosome that encodes for a transcription factor. And so we have transcription factor A that comes in and docks on, again, a cis-acting element, a part of the DNA along the same chromosome relative to our gene of interest here. In response to and so that's going to be true in both cases here. In response to stimulus B, we'll have another transacting element that encodes for a transcription factor that might only bind to, that might also bind to the same gene, but might not bind to another. And so, again, we have one transcription factor A, that can bind to different genes, but we also might have multiple that bind along one single gene here. And so with more transcription factors bound to this gene on the left, how might that affect the transcription of this gene? Will it lead to more transcription, less transcription, or will it just be the same? I'm seeing small gestures of up, which is correct. More transcription factors will lead to more transcription of that gene. So it more readily recruits RNA Pol II, leads to transcription, whereas fewer transcription factors on that other gene will lead to less transcription. But again, there's this, it's not again, as someone asked earlier, this isn't a yes, no, off on binary dynamic, but rather a degree of transcription. There's a higher degree of transcription with more transcription factors. If there's only a single, for example, transcription factor here, that's part of the initiation complex, and there's still going to be transcription. It's just going to happen to a less degree. Let's see a couple of hands. Yes. Is that the next slide? Hold on, give me 1 second. Yes. And there's a great slide that I have that kind of helps illustrate that, where there's like markings along a gene where things can bind. But yes, multiple transcription factors can come in and dock and bind to at the same like cis-acting element. Yes. More transcription. So RNA Pol II will more readily be recruited to the promoter and promote transcription. And so transcription is typically not, and maybe this isn't something I explicitly said or didn't do a good job of articulating. When we talk about transcription, it's not a single reading event, but it happens multiple times. And so the longer a transcription factor is there and it's bound, the more readily transcription will keep happening along that gene, producing more and more mRNA transcripts. When we talk about gene expression or measuring gene expression, we typically do so by measuring the amount of mRNA, amount of mRNA associated with that gene. Does that make sense? One-to-one. With every transcription event, there's going to be one mRNA strand produced. Yes. Yep. That's exactly right. All right, we're going to press on because I want to make sure I don't go over on time. So when talking about these cis-acting elements or these cis elements, we talked about how this is typically along the same chromosome. There are different categories of cis-acting elements. There are those that are what we dub as proximal cis-acting elements or proximal control elements. Those are typically those that are about 100 to 200 base pairs upstream or downstream from the promoter of a gene. And those that are distal, we typically say that are more than 100 or 200 base pairs away. Typically, they are thousands of base pairs away. It's that level of magnitude where proximal elements are usually within the hundreds, like 100 to 200. Excuse me, base pairs away, but the distal elements are usually thousands of base pairs away. For the distal elements, that's what's depicted here. Proximal is not depicted on this slide. For the distal elements, the way they work is that they can actually either enhance or repress transcription. And if it enhances, those distal elements are usually referred to as an enhancer. What it does is that they will, again, our DNA exists in 3 space. It's not just a single strand. There's folding of our chromosomes so that they're compact and able to be packaged within our nucleus. And if you think about the 3-dimensional folding and space of our DNA here of a single chromosome, there's going to be distal elements that might be close or adjacent to some distal gene. And so there are some regions of our DNA that have been identified as enhancers that might be extragenic DNA, might not be a coding region of the DNA, however, has been identified as a region that promotes and enhances transcription at a specific gene. And so this bending over of our DNA allows this distal site where proteins will bind to it, creating what we call the enhancer zone. that more readily enables transcription to occur at some given gene of interest. On the opposite end of that, there are those that can block transcription from occurring, block transcription factors from binding, and inhibit RNA polymerase from binding to that gene of interest. Those are repressors. And so they're inhibiting or repressing expression of that gene of interest. And so one group from our department here looked at the known consensus sequence for what these transcription factors, FOXO and RUNX here, what cis-acting elements they bind to, and we're able to look at different genes and see on those genes where these transcription factors are able to bind to. Typically, when looking at this, when looking at genes illustrated in this way, this right angle arrow typically refers to the start site for transcription. And so we typically will have these transcription factors binding to the promoter region just upstream of this start site. But as you can see, these transcription factors can bind all along a given gene, and typically will help facilitate and enable transcription here. But as you can see, primarily they're going to be upstream of the start site in the core promoter of a given gene, where the transcription factors will bind and have their consensus sequence or the cis-acting element that they recognize and bind to. Again, transcription factors have DNA that they recognize. They will typically bind to other proteins, but will be a unique class of proteins that can recognize specific sequences of DNA, bind to that sequence of DNA that they're destined to recognize, and promote transcription of some given gene that possesses that sequence. And again, UniPro, if in your spare time you want to look at DNA sequences and look at what genes transcription factors bind to, UniPro is a great website to just pick a transcription factor and you can look it up. If you want one for inspiration, STAT5 would be a great one. Yes, go ahead. Is there an instance where a transacting element is closer than a cis-acting element? Well, no, because a distal control element is still a cis-acting element. That means it's on the same chromosome, where typically a transacting element is going to be on a totally different chromosome, typically. Does that make sense? And so if you, for example, this is a great exercise. If you, again, want to spend your Friday night looking at UniPro, you can look at what chromosome the transcription factor you're interested in is located on, and then what the genes that it shares that they regulate or bind to, what chromosomes those are on. And they're typically going to be on totally different chromosomes, which means that transcription factor is a transacting element on a totally different chromosome than the gene it actually binds to. Does that make sense? We have about 5-6 minutes. I think I can get us there. I'll apologize ahead of time if I don't. So bringing this back to diabetes, we have a few different forms of MODY that have been identified. One particular form of MODY has been associated with an issue in transcriptional regulation, and so it's this 70% genotype that's associated with HNF1 alpha. So MODY and HNF. So the key players here are HNF, which is hepatic nuclear factor, and DCOH, which is dimerization cofactor of homeodomains. From here, we're just going to refer to them as DCOH and HNF. Okay, what ends up happening is that HNF is a transcription factor that can bind to the DNA when it's in a dimer form. Okay. And DCOH is a protein that's required for certain transcription factors, including HNF here, that associate with this transcriptional machinery. And so HNF requires DCOH to help contribute to the formation of this initiation complex. And HNF requires DCOH to actually bind to the DNA. What this looks like is that DCOH is typically in a homotetramer, same subunits, tetramer meaning 4, And so HNF is typically going to be in a monomer form, but needing to, in order for it to function as a transcription factor, it A, needs to be in a dimer, and it needs DCOH to help it form that dimer. So what ends up happening is that HNF and DCOH will associate together to form this heterotetramer with 2 HNF units and 2 DCOH units. And it's now able to bind to the DNA. And where it binds is on the insulin gene. It'll bind at the promoter, promote transcription of insulin. And so you'll get insulin mRNA, which is then encoding for the insulin protein itself. What ends up happening in Modi is that there are hundreds of mutations in HNF that have been associated. Of those mutations, some of them have been identified to alter the ability of HNF's dimerization with DCOH. So in Modi, we're not able to have HNF is unable to form its dimer with DCOH. And what that leads to is the inability for HNF and DCOH to transcribe and promote expression of insulin. And so we have a ribbon protein structure illustration here for DCOH and HNF here. And again, it's a mutation in HNF that enables it to dimerize with DCOH. The treatments for Modi that possess these HNF mutations, as you can imagine, insulin treatment. We have also oral hypoglycemics, sulfonylureas that actually promote insulin release by indirect means. It's not leading to more expression, but more readily shuttling it out of the organelles of the insulin that is being expressed. We also have biguanides that lead to inhibiting glucose release from the liver to try and help, again, promote hypoglycemia, lower blood sugar level. There are more experimental approaches that are being pursued now. Pancreatic islet transplants that take the islets, these like functional units of the pancreas that possess the beta cells, and transplant them. There's some ongoing issues with that, but it's an ongoing area of investigation to improve this cell-based therapy. And there's also whole pancreas transplant as well. That again, it's an area of active research as well. For steroids, we're almost there, and I think I'll get us done by 3:15. For pivoting to anabolic steroids, steroids are typically a class of molecules that share this common structure. So they have these 3 6-carbon rings with a 5-carbon ring to the far right. This is typically called the steroid nucleus of a steroid. And so every steroid will have that same common structure, whether we're talking about testosterone, estrogen, progesterone, or corticosteroids. And they're typically incredibly hydrophobic because of this hydrocarbon structure. And because of this hydrophobic nature, these steroids that are typically endocrine hormones that are transported through the blood need to be shuttled through a carrier protein to address this hydrophobic nature. And so one of the most abundant proteins in our blood is called albumin that acts as a carrier protein to shuttle hydrophobic hormones or hydrophobic steroids throughout the body. And typically because of how hydrophobic these steroids are, they're actually able to cross the cell membrane on their own, not needing a transporter or some sort of shuttle to do so. And they'll shuttle across the membrane, and the receptors for these hormones are actually intracellular. They're inside the cell. What this looks like is typically after this steroid hormone passes through the cell, and in this case, it's going to be cortisol, the steroid receptor, in this case, glucocorticoid receptor, has this inhibitor protein on it. When the steroid binds to its receptor, it will promote the release of this inhibitor protein. And now it's actually able, this receptor is now actually able to act as a transcription factor itself. So what happens is that 2 of these hormone receptors will dimerize together, form this complex that can actually clamp onto the DNA and will promote expression of some gene that possesses a glucocorticoid response element. And so that's going to be the site at which the glucocorticoid receptors will bind to and will promote transcription. An example of this is a synthetic steroid anadrol. That's a synthetic testosterone. It will typically bind to the androgen receptor. That's the testosterone receptor present in our bodies. And so that this anadrol is typically an anabolic steroid that will end up binding to this receptor. And what actually happens is that anadrol is able to bind more specifically to androgen receptor than compared to natural testosterone. And it does so more strongly and is typically unable to let go of that androgen receptor. So when it binds more readily, that means it's going to stay in its active form, serve as a transcription factor, and keep expressing the proteins that it identifies, or the genes that it identifies and the proteins that go along with it. It also prevents some post-translational modifications of the receptor, meaning that it is less likely to stop phosphorylation, maintaining being in an active form. And so these are the differences between natural testosterone and synthetic forms like anadrol that lead to overactivity of these testosterone-associated pathways that are associated with the negative side effects of anabolic steroid use. For the genes that are associated with androgen receptor transcription factors are going to be IGF, insulin-like growth factor. This is a growth factor that, when it binds to the receptor, activates a series of protein synthesis pathways, cell growth pathways, and actually inhibits protein degradation. So it triggers a lot of anabolic processes associated with protein synthesis. It's actually, IGF-1 is used therapeutically to treat those that have growth issues. And so particularly those who are prepubescent or have halted or stunted growth. And IGF-1 is also used as a performance enhancing agent. So in addition to compounds like anadrol, IGF-1 is also on the kits that they test for when testing athletes for performance enhancing drugs. IGF-1 is also what helps distinguish between the differences in animals. So for Great Danes, it's been identified that they have far more IGF-1 expression compared to a Chihuahua that has far less IGF-1 expression. So it's also associated with difference in size. And so with that being said, if transcriptional regulation fascinated you, here's some folks at UVA that are in this space. I kept you longer than I promised. Forgive me for that, but thanks for sticking around. Yep.